Hello everyone, welcome back to PhytoPathoVerse. I'm Malini, molecular plant pathologist with the motto to make plant pathology easy. Today we will move forward with chapter 10 GN Agrios, Control of Plant Diseases Part 2. In the last session, Part 1, we have covered the agronomical and the biosecurity methods to manage our plant diseases, and in today's session we will see how crop protection products play an important role, and the genetics, the way of the genetics and plant breeders, how they help us in managing plant diseases. And there are many more, so we will see all of those in today's session. Starting with genetics, using disease-resistant crop varieties. It is frequently stated that the use of resistant varieties is the least expensive, easiest, safest, and most effective means of controlling plant diseases in crops. This is certainly true under many conditions, but it should also be noted that genetic disease resistance generally provides only partial protection against one or few pathogens, that the resistance can break down in a few seasons, requiring replacement of the cultivar, and often comes with significant trade-offs in terms of reduced yield and quality. Partial resistance. Nowadays we have many technologies such as MAS, but previously it used to take 7 to 8 years, as of how I learned. So it takes that much year to come up with a new variety, but with the coming technologies it takes few years. That's fine, but what happens, they cannot give us total protection. They give us only partial protection against one or few pathogens, because that resistant variety is made targeting a particular pathogen or a group of pathogens. What about the rest of the pathogens, right? So yes, they give partial protection, not total. Disease resistance is normally one of the top 3 or 4 goals of the plant breeder. Although yield and product quality may well have higher priority, breeders will normally make substantial attempts to provide an adequate resistance package in their varieties. This is a major challenge because the breeder needs to find ways to improve disease resistance, not just to 1 but up to 20 pathogens at the same time as maintaining all the other phenotypes of interest. Now this is a challenging thing for the breeders. Now their first priority is yield and the product quality, because again the yield and the product that will come out of that crop is essential for commercial trades and commercial sale. Okay, now that is their first top priority, but along with that they need to keep in their mind how to find ways to improve the disease resistance. Okay, and not only just for 1 or 2 pathogens but up to 20 pathogens. Okay, this is an assumption, up to 20 pathogens, which they mean is that they should or they find a way in which that resistant variety should target more pathogens rather than the fewer ones. So they have to keep everything in their mind while coming up with a new variety. The genetic basis of disease resistance has been studied for over a century and has progressed massively in the last 20 years. Genes conferring resistance to pathogens were first mapped and then cloned, showing that most encoded NBS, LRR and PRR proteins, we have already seen about this. They have also mentioned, it has been covered in chapter 4, so you can go and see that. The genomes of most major crops and the model species like Arabidopsis have been sequenced, showing that several hundred NBS, LRR and PRR genes exist in the genomes. Converting this information into useful tools for plant breeders is a substantial task. The most obvious route are to use molecular genetics to move a disease resistance gene into a cultivar that is susceptible. This GMO, genetically modified organisms route, will surely become normalized in the not too distant future, but so far has not resulted in a field application. More common is to use molecular markers linked to resistant genes to assist breeding operations. Okay, now what are the sources of these resistance genes? A plant breeder faced with a cultivar that is unacceptably susceptible to one or more diseases must initiate a search for a source of useful resistance. The options are: the first option, elite cultivars adapted to the local condition and market segments. This would be obtained from the breeder's own stock and from the other breeders. Okay, this was the first option, the elite cultivars. So once released, breeders can use the cultivars for competitor companies. Okay, the second one, any other lines of the same crop including land races. Global collections of cultivars are maintained by national authorities, research institutes, universities and the CGIAR institutes. Importation of these lines from other countries and regions may well be subject to quarantine regulations and may take months or years to complete. This was the second source of the resource resistance genes. Okay, third is wild relatives of the crop species. Collections of wild relatives are held in national collections but are subject to the Cartagena Protocol Convention on Biological Diversity. I believe this wild relatives of species means the ones which are familiar with this, which are familiar and they have their natural tendency to defend to such pathogen. With those wild relatives, our cultivated crops can be crossed, or with the help of the wild relatives, the new variety can be developed. Means what are the reasons because of which gene or anything else, because of what parameter these wild relatives are resistant naturally to the pathogens and our cultivated crops are not. So they find out the source and they try to incorporate it in our cultivated crops with coming up with a new variety. Number 4, any of the above lines can be used as the starting material and subjected to a mutagen. Example, UV, gamma rays, EMS, genomic edited or transgenetically modified. Now the next step is to screen the assembled lines for resistance to the troublesome pathogen. The most obvious route is to grow out the lines in a test plot under farming conditions and wait for natural collection to occur. Now what they will do screening of the okay, now they have potential resistance genes. They have collected potential resistance genes from all the 10 sources, for example. Now what they will do, they will test it. Like we have lab testing, field testing, whatever the thing, they will screen it. Screen it means what, we have sieve in our homes, so like that. So they will test it, which gene is working best to resist that pathogen. So that's how screening is done. With replications and various composition mixed, they will do screening. Okay, that's what they have mentioned here. Yeah, you can read. Okay. Now see the same thing, first they will screen it on their own field and then they see. The final test will be whether farmers find that variety gives them better yield and quality than before, and whether the cultivar remains resistant for enough time to recoup the investment made by the breeder. So that is the thing. First the breeder will find the source, and they will do the lab trial, field trial, and then they have to do on farm trial. So the farmers will be motivated to use that variety. So then the farmers will check whether that new variety is giving them more yield or not, and then finally they will come up with a conclusion. They have also given a case study Okay, on this, moving further, the next is impact of genetic disease resistance. What is the impact? The use of varieties resistant to pathogen has had a major impact, especially in crops that are grown as annuals or biennials. Genetic resistance has been found against all major types of pathogens, fungi, oomycetes, viruses, bacteria, and nematodes. In some cases, resistance is very marked, giving immunity from the pathogen, especially to biotrophic and hemibiotropic pathogens. In other cases, especially the broad host range necrotrophic disease, significant genetic resistance is very rare. The ability to breed crops for resistance is directly related to the generation time and seed production. Genetic resistance is a much more difficult proposition for perennial crops such as fruits and forest trees. The long generation time is the key consideration. However, some apple varieties resistant to apple scab are now available. Examples: forest trees disease managed with resistance are white pine blister rust and fusiform rust of pine. So these are the impacts of genetic disease resistance. Next, maximizing the lifespan of the disease resistant cultivars. How they can increase or maximize the lifespan of these cultivars. So having gone to the effort of breeding a new cultivar with adequate disease resistance, the next challenge is to maximize the effective lifespan. Several related strategies have been adopted. Let's see what are those. Using weak rather than strong resistance gene, using recessive rather than dominant recessive gene, using many rather than a single resistance gene, which is stacking. Okay, till now they are talking about a type of horizontal resistance rather than vertical. Okay, next, using different cultivars with different resistance profile, gene profile in the same field, in the neighboring fields, or in successive seasons. Monitoring for the breakdown of disease resistance genes, limiting the pathogen population using fungicides, crop hygiene. So these are the tactics that can be used or that they are using to maximize the lifespan of the disease resistant cultivars. So we will see one by one what they are trying to say. First, weak resistance genes. Breeders and researchers have often worked on strong resistance genes that give immunity or at least very low disease levels. However, even though they are easy to detect, map, and integrate because they have a large and clear effect, they have repeatedly been shown to break down quickly. Again, now in this 4 to 5 lines, they are saying, first of all, means initially the breeders and researchers, they worked on strong resistance gene, one or few strong or major genes, resistance gene. Now the techniques used for such type of genes is vertical resistance, in which the major and the strong genes are used, but in the limitation, the breakdown means in the limitation, the main setback is they break down quickly because we have one target gene. So pathogens are also clever, so they will find a way to break down that one or two genes. But nowadays what the breeders are doing, they are incorporating many minor genes all together so that the pathogen get confused, or the pathogen will get tired which one to break. 10, 20, 30, if they will stack it all together, so it is not possible for a pathogen or anybody to break down. There is a saying, unity is strength. So that is it. The rationale means the objective is that if a gene can be overcome by a single mutation in the pathogen, there is very high chance that this mutation will be present. If not in the field itself, then in the locality or region. Furthermore, if the mutated pathogen can grow rapidly on the new cultivar, the selection pressure is correspondingly high, leading to a rapid growth of the pathogen and resistance gene breakdown. Same thing they have mentioned. Since although weak resistance genes that permit some pathogen growth are harder to work with, they provide longer term protection. So the horizontal resistance is more durable than the vertical ones. We know that. The next point is recessive gene resistance genes. The classical model of NBS-LRR disease resistance is that the plant recognizes an effector molecule from the pathogen, leading to an effective defense response. Again, you can see this in chapter 5, what they are saying, such as effector, the NBS-LRR. In chapter 4, 5, they will, you can see in chapter 5, if I'm not wrong, the chapter 5 is about plant immunity. So in that, every term has been explained. If the pathogen effector gene mutates, the pathogen may evade detection and cause disease. Such dominant resistance genes are inherently sensitive to breakdown. If instead disease requires the active participation of a host gene, the resistance gene is recessive and conversely susceptibility is dominant. Good examples are the barley powdery mildew disease gene MLO and the wheat TSNL and SNNL gene. The durability of MLO indicates that there are no straightforward ways for the pathogen to evolve and evade such a recessive resistance gene. The next is multiple resistance gene, in which the stacking will come. If a host plant carries two independent resistance genes, the pathogen must mutate both. That's possible. They can mutate both correspondingly effector genes to cause disease. Now, what will happen if there are many more? Such as mutation of one effector does not confer virulence on cultivar carrying another effective R genes, and so would not be selected in the population. Furthermore, the chance of both effector genes simultaneously mutating in the product of the chance of each gene mutating individually. Hence, the concept of using two or more independently effective gene resistance has gained much attention. So, we can use more resistance gene altogether. This is especially the case for high value genes such as R genes generated using GM technology. However, there are some severe difficulties with the assembling and deploying, such as gene stack. One problem is that if the genes are present at different locations in the genome, breeding with such a cultivar while retaining all the desired R gene is very cumbersome. Using GM techniques, it is feasible to place all the genes at the same locus. A second issue is that all the genes should only be used as part of the stack and not individually in any other cultivar. Yes, they should be used on a stack, not individually. Any use of single R gene risk pathogen evolving virulence to one gene at a time. I hope that's clear. If there will be a single gene alone, and it will be defeated by the pathogen. If all the genes will be all together in a stack, a group of genes, then the pathogen will be scared of those genes, a group of genes, right? If any gene is defeated, this gives the breeder the early warning that this gene must be replaced by the stack. Okay. The next is cultivar mixes. Monocultures are inherently susceptible to disease and resistant gene breakdown because all neighboring plants are also susceptible to a pathogen that has acquired virulence on the cultivar. These disadvantages of monoculture can be reduced by sowing a mixture of cultivars that carry different resistance genes, sometimes called varietal mixtures or multilines. So now you know what are varietal mixtures or multilines, which means the use or the sowing or the cultivation of the cultivation of crops having different resistance genes. Okay. Spores from pathogen sowing on a susceptible cultivar are likely to encounter resistance cultivars during spread. This reduces the rate of epidemic spread. All the results are variable. Cultivar mixes suffer less from disease and may have greater yields. One disadvantage of cultivar mixes relates to agronomy. What is this disadvantage? It can be difficult to find two cultivars that have identical growth characteristics and thus can be sown, cultivated and harvested simultaneously. The other difficulty, okay, this is one difficulty, fine. One. What is another? The other difficulty relates to product use. If cultivars are destined for specific uses in product chains such as baking or brewing, manufacturers may well find it difficult to use a mixed input. That's true. Mixture, their qualities, their factors, characters will not be identical, so it will be difficult for the manufacturer to use it as an input. Mixtures are well suited if the product is used as animal feed. Okay. Some of the disadvantages of cultivar mixes can be accurate by growing different cultivars in different rows fields in a successive seasons. In these cases, the agronomic difficulties are reduced and product uses can be kept separate. The next is monitoring of resistance gene breakdown. An outbreak of a disease may well be restricted to a small region at first. Epidemiologically, principles can be applied to predict the rate and direction of spread. Therefore, opportunity exists to warn farmers of the likely appearance of a virulent pathogen in the coming season. An example of this occurred in the Eyre Peninsula region of Australia, where canola cultivar with major gene resistance to blackleg are grown. This some example they have given here again. So technologies to screen pathogen population with genotypic markers are now very efficient and can be used to screen for the presence of virulent genes. Such technologies may well allow a more dynamic approach to deploying resistant genes in cultivars as threat from pathogens emerged and recede. Next is limiting the pathogen population using fungicide and crop hygiene. The chances of a mutant population emerging and evolving that can overcome the resistance gene in a new cultivar. That will always be greater if the pathogen population is larger. You would be expected that uncontrolled epidemics of any pathogen will threaten the longevity of any resistance gene in cultivars. Other means to control disease include CPPs, especially fungicides, crop rotation and crop hygiene. Using all these methods is predicted to prolong the effective life of disease resistance gene. This is integrated pest management. The next point or the topic of after this will be disease resistance via genetic modification. So now technologies to move genes into crops species were developed first in 1970s and that have been developed and refined continuously since. Nearly all annual and biennial crop species can be transformed and even some perennial and woody species. When first developed, the methods were established for a particular crop variety that may no longer be commercial cultivation. Nowadays, methods that work on any cultivar have been published for many crops. This development stimulated a major expansion in plant pathology research from the 1980s and brought into the subject people trained in genetics and molecular biology on other systems including myself. The potential to transfer gene conferring disease resistance from any source into crop plant promised to revolutionize the discipline as it would free breeder from the constraints of conventional crossing based system and free farmers from using pesticides and unfavorable rotation. Okay. For the next 2 decades, scientists labored to map, identify and clone the genes to transfer into crop plants. A small number of transgenic crops were successfully developed expressing genes from herbicide tolerance, insect tolerance and Harvestability. The tickle of clone disease resistance gene became a flood around 1998 just as the market for new genetically modified crops abruptly closed. A range of concerns such as human health, environmental impact and multinational company involvement were cited by those opposed GM crops. Okay, moving forward. More recently, genome editing techniques have been developed for many crops. The ability to specifically alter genes was first developed by zinc finger nuclease and transcription activator like effector nuclease, which is TALEN, but it is widely predicted that clustered regulatory interspaced short palindromic repeats CRISPR-Cas. See, this is the full form of CRISPR and this is the full form of TALEN. For TALEN, it's transcription activator like effector nuclease. For CRISPR, it is clustered regularly interspaced short palindromic repeats. Okay, will prove revolutionary for crop disease control because it enables a cheap method to alter genes in all crop species. This will enable the modification of NLR disease resistance genes so that they detect a design suite of pathogen isolates. Secondly, many crop genes such as MLO that confer susceptibility to pathogen have been discovered and these are obvious candidates for disruption by GE techniques. As GE techniques do not involve integration of new DNA in the recipient, some regulatory authorities have sought to distinguish GE from GM techniques and apply much less stringent test similar to that applied to conventionally bred crops. The next is genes from plant virus that interfere with viral replication by inducing gene silencing. As plant viruses are small genomes, their complete DNA sequences were obtained as early as 1980s. For reasons that were not immediately clear, expression of viral coat proteins gene in plants was shown to confer resistance to that specific virus. The first such case was tobacco transformed with the coat protein gene of the tobacco mosaic virus, which then became resistant. Other cases include squash transformed with the coat protein genome of cucumber mosaic, squash mosaic and watermelon mosaic papaya virus, and papaya transformed with the coat protein gene and papaya ring spot. Now the thing is, in viruses there are only two things. One is coat protein, which helps. Now let me first tell. The first element is the nucleic acid, which is our DNA or RNA, and the second one is the protein coat, right? So these are the two things with which the alteration can be done, with which all the experiments can be done, because they do not have any other organelles or any other thing within them. They just have nucleic acid and protein coat. Okay. The next is NPS LRR disease resistance genes from another cultivar while relatives or species. While relatives of crops often carry resistance genes that are missing in current cultivars. Modern genomic technologies have made it relatively straightforward to clone such genes. Gene expression bypass by the difficulties that would result from programs to integrate such genes into current cultivars by conventional crossing method. A recent case addresses the chronic problem of potato late blight. 3 resistance genes from the close relative of potato, Solanum venturi and Solanum americanum, were transferred into the current commercial potato cultivar, Maris Piper. In field trials, the GM lines were resistant to all tested strains of Phytophthora infestans. The commercial release of these varieties is now dependent on regulatory authorities. The next is antimicrobial proteins. A wide range of protein exhibit general antifungal properties. Some such as the PR protein family. PR means pathogenicity related family. We have seen there are various families, 17 families. Okay, and their functions in previous chapter, right? So other example include tomatins and osmotins, chitinase proteins degrade the fungal cell wall and release chitin fragments that potentiate the plant's defense response. Defensins are a diverse class of macrocyclic proteins that appear to create pores in fungal membranes. So, okay, fine. Next is antimicrobial metabolites. Many of the specialized metabolites produced by plants have antimicrobial activities and may have a role in disease resistance. Some compounds are present regardless of infection and are known as phytoanticipants. Others that are induced in response to infection are called phytoalexins. We know the difference between phytoanticipants and phytoalexins. I'm not going further with this. The biosynthetic pathway to such compounds are normally complex with multiple genes and highly regulated pathways. Despite the potential complexity, GM approaches to enhancing the SM content of plant has been the subject of much research. In a few cases, the expression of a single gene can enhance the production of an antifungal product. One widely publicized case is a stilbene synthase which produced the stilbene phytoalexin resveratrol. The gene from grapevines was transformed into at least a dozen plant species and gave broad spectrum resistance to a wide range of biotrophic, hemibiotropic and necrotrophic resistance. Furthermore, the antioxidant properties of resveratrol were claimed to improve nutritional quality. However, such plants have not been commercialized so far. Next, disruption of susceptibility genes. Okay. A surprising number of disease resistance genes are genetically recessive. This means the active gene product confers susceptibility to a pathogen. The best known example is the barley powdery mildew gene. Random mutagenesis was used to create inactive amino alleles and this have been incorporated in a successful series of cultivars. Similar genes exist in many plants and genome editing is now an attractive method for inactivation, especially when the gene is in a polyploid species. Many other cases of susceptibility genes have been found, often by mutagenizing plants and screening for disease resistance. Once found in the species, orthologous genes can be sought in crops and inactivated by GE technologies. The next is host-induced gene silencing. The ability of RNA molecules produced by plants to interfere with gene expression by the pathogen cells has many attractive features. Such host-induced gene silencing is an attractive concept. What it does, the plant are engineered to produce RNA molecule, yes, that can cross all cell walls and membranes and interfere with the gene expression. The RNA molecule can be chosen so that it is highly specific to a key housekeeping genes of the pathogen. Negative impact on non-target species seem very unlikely. One example seeks to suppress the expression of CYP51 gene, which is also the target of DMI fungicide. Another case, DMI, DMI is the demethylation. I think it will be mentioned somewhere. Let's see if we move back. Okay, where is it? Fine, I cannot find it right now, but okay, DMI is demethylation. Okay. Another case seeks to suppress the expression of genes used by fungi to synthesize mycotoxins. A related approach is to synthesize the RNA and spray it on plants like a fungicide. Spray-induced gene silencing. These concepts are being much researched and seem very promising. The next is crop protection products. Okay, till now in today's session, till now we have seen about the genetical approach for managing plant diseases. In the last session, we have seen the biosecurity and agronomical approach to manage plant diseases. And now this is the last point, but we have many more to know, but this is the last point or concept in which plant disease is managed, which is crop protection products. In crop protection products, you can see there are many things, right? Let's see. Even when the best biosecurity procedures have been followed, the most disease resistant cultivars have been used and efforts to prepare the field have been undertaken. Decreases normally affect crops. If the threat of the pathogen is high, the only option left to the farmer is to purchase and apply crop protection products. Yes, right. One or more crop protection products. So CPPs can be divided into 8 classes and many products have roles in 2 or more of these classes. So what are those 8 classes? Fungicides, insecticides, arachicides. Arachicides means the chemicals that kills mites, spider and mites. Nematicides kills nematodes. Insecticides, fungicides, I hope you understood. Antibiotics for bacteria, herbicides for herbs, plant growth regulators, and rodenticides that kill rodents, right? Rats and all, okay? So CPPs are tightly regulated by national and supranational authorities that also divide CPP into product categories. Now, what are those categories? One is basic substance, then synthetic molecules, then botanicals, and the living organisms. So what are those? The basic substance is the simple chemical that have been used for many decades and are generally regulated as safe. Can you give me one such example? I know it will come in the subsequent paragraph, but if you know, that is better. So I'll give one example such as sulfur. Sulfur can be used as basic substance, or copper maybe, okay? Then synthetic molecules, there are synthetic chemicals, botanicals, neems and all, they are the botanicals. So single molecules and mixtures extracted from plants and other sources but not subjected to deliberate chemical modification. And the last one is living organisms such as biocontrol agents. Now we are concerned here with the case of CPPs to control disease of crops and other plants. By far the most important class of CPPs are the synthetic fungicides. This one, synthetic molecules, synthetic fungicides. Now why they are saying that? Because in the fungicides or in chemicals, there are 2 ways again divided. It can be divided into local or contact fungicide and systemic fungicide, okay? And here they have talked about synthetic. I'm really sorry, I got mixed up with synthetic and systemic. Leave it. Moving forward, but basic substances, botanicals and biocontrol agents are important and in some sectors are growing parts of the market. Antibiotics and insecticides have smaller roles. We describe here some of the most important classes of CPPs. The organic agriculture movement, also known as biological, biodynamic and many other names, permits the use of CPPs except those that are synthetic. Values, let's see, we will be cleared with each and every words that have been mentioned or the classification that have been done. Values of CPPs. The total annual sales of fungicides are now exceed $20 billion and is increasing at about 5% each year. However, the total weight of products being sold is slowly declining, reflecting the greater activity of newer products. Decisions to purchase and use fungicides are taken directly by farmers and are rarely subsidized or otherwise encouraged, so farmers are clearly convinced that the cost of fungicide purchase and the application are paid back by increased yield security and crop quality. Pesticide companies and farmer-funded groups undertake efficacy trials to check that fungicides are effective and to determine optimum doses and application methods. The degree of payback varies accordingly to the cost of the fungicide and depends on the amount of the disease and the value of the harvested crop. The cost of the applied fungicide varies enormously. In broad acre crops, the cost of the product for each application can be as little as $5 to as much as $1,000 per hectare. Okay. They have discussed about the values of the commercial purchase. Fine. Next is approval of fungicide. Regulation governing the sales of pesticides were substantially reformed after the negative impact of organochlorine insecticides was recognized in 1960s. Registration is governed by national organization with the American EPA and EU authorities being partially influential. The cost of registration is about $200 million per active, representing the largest part in the development of a fungicide product. There has also been a general tightening of regulation so that existing pesticides are subjected to re-evaluations and many are deregistered, especially when new and better products become available. The criteria used to judge the safety of a pesticide vary between countries, but all are designed to demonstrate that they are effective, do not harm the user or member of the public, and do not harm non-target organism in the environment. All pesticides are toxins and there is a clear need to encourage the use of the smallest dose. To achieve this, some countries such as Denmark have added an additional tax to pesticides to discourage the more toxic product. That is a good way of not using much pesticides. Okay, the pesticides is scored for a range of toxic properties relevant to Danish conditions, impact on human health, ecotoxicology and environmental fate. These impacts are converted into a tax, so a farmer needs to weigh up the extra cost of a fungicide versus the control that a particular compound affords. Fungicide regulations are summarized in product labels that govern the way a product can be used. Such labels specify the crop and often the disease, place upper and lower limits on the dose, and limit the number of sprays that can be given in a season. They often describe whether the product can be mixed with other CPP's fertilizer. Finally, it describes the withholding period. This is the minimum period between the final spray and the harvesting date. Next, we will see the types of fungicide used. MOA means the mode of action, spectrum, resistance, preventive, curative, contact and systemic. Now they have used that term, contact and systemic. Previously, it was I got confused between systemic and the synthetic. Not confused by the definition but with the word. That's what I'm saying. Now let's see what all this word means. First of all, mode of action, the biochemical process that this CPP inhibits to reduce pathogen growth. That means what? There are many fungicide groups. So what are their target site or target location that they are saying, with the help of which they decrease the pathogen growth? Let me give you one example. Like there are groups such as oxathins, strobilurins, and then benzimidazole, right? So all are there. Then triazole. So all this, for example, I have given 4 group names, right? But the 4 group names, in each group there can be many chemicals, such as in oxathin group, 2 well-known chemicals are there, which is carboxin and oxycarboxin, Vitavex and Plantvax, right? Why is it famous? Not famous, well known. Why? Because carboxin is the first synthetic fungicide reported by which scientist? This is a question in this session. I have not asked any question, so this will be the first question to be asked. Okay, which is the scientist who has developed the first synthetic fungicide, which is carboxin? It's not that tough. So what I was saying is that so carboxin group, their mode of action is SDHI, succinase dehydrogenase inhibitor, right? Whereas for triazole, the mode of action, they target the sterol biosynthesis. Fine. So these are the mode of action. I hope we will there will be a table given, so you will understand. So that is the mode of action with the help of which they inhibit and reduce the growth of pathogen. Next is spectrum, the range of host disease combinations controlled by a CPP. Yes. Okay, the range of host disease combination controlled by a CPP. So what is the host range? It can be small host range, or the chemical can be broad spectrum. Broad spectrum means the chemical can target multiple host, right? So that is the spectrum. The third is resistance, evolved and heritable decrease in the sensitivity of a pathogen to a CPP. So resistance, you know, obviously. Preventive with the term only you can say. The chemicals that are applied prior. Preventive means prior, curative means after. Okay, that is applied after the infection. The next is eradicant. Eradicant we have seen in the last session also. Eradication. Eradicant means they have established pathogen and they want to kill it or eradicate it. Contact and systemic. Contact means they will have direct contact with the pathogen. Okay, systemic means they have to move to that location. They have to travel. They have to travel within the plant and control disease remotely from the point of application. Means application can be in the root, for example, I'm saying, or leaf, not root. Application can be, why root? Root means application in the soil application I meant to say. Or you can say the chemicals can be applied on leaves, like the foliar application. So in case of contact, what they have to do, they have to spray on the specific location where the disease has infected, so that they can have a direct contact with the pathogen whatsoever. Now the contact fungicides, you know, they have multiple side of action. They don't have a single site. So if the disease is here in leaf or in stem or somewhere else, so they have to come to it directly to be effective. But in case of systemic fungicide, they have single side of action. Yes, so single side of action, why? Because, you know, they only target specific side. The example that I have given you while describing about the mode of action, for oxathane it's SDHI, for triazole it's sterol, for strobilurin is quinone, cytochrome B. So they have single side of action. Now for that, what happens, this systemic fungicide can be break down easily, because, you know, single side of action, whereas contact fungicide, they are multi side of action. So every chemicals, they have pros and cons. But in contact, they cannot move. They needs to be directly touched with the pathogen, whereas systemic fungicide, they can move to that location, right? They can travel to that location. Moving forward, active ingredient, the active molecule in the product which also contains solvents, adjuvants and the emulsifier. The commercial product name may not be related to the AI name. AI means what? Artificial intelligence? No. In this case, AI means active ingredient and may differ in different countries. Okay. Now we will see what are the basic substances. Before some time we have seen this differentiation, right? Basic substances, synthetic, botanicals and living organisms. So here we are starting with basic substances. So the example I have given you was sulfur, as far as I remember. So the example can be sulfur and copper as basic substances. Now the history of using defined chemicals to control disease goes back to the 18th century and predates by a century the widespread recognition that diseases in animal as well as plants were caused by specific microorganisms. Matthew Tillett examined the efficacy of various treatments against cereal, burnt and smut diseases, showing that lime or ammonia was effective. Excuse me, sorry. Now history section has already done, but as I got a scientific name, I have to ask you. Matthew Tillett is well known for what? He is the father of what? Right? Father or something else. It can be something else. Okay? So go and check Matthew Tillett is called what? So by 1855, Becquerel was using powdered sulfur to control grape powdery mildew. By 1885, a mix of lime and copper sulfate, known ever since as the Bordeaux mixture, was developed by Millardet and was shown to control not only grape downy mildew but also late blight in potatoes. Several versions of the treatment were explored, but the mixture developed then is still in use today for the control of fungal and oomycete disease on a wide range of crops. Under EU legislation, copper and sulfur was called basic substances, a diverse grouping that currently includes a range of inorganic and organic materials. Their long history of apparently safe use means they are subject to a less stringent regulatory framework than other CPPs and are permitted in organic system in some but not all countries. The major basic substance fungicides are based on sulfur and copper. Now coming to the mode of action of this copper and sulfur. So the mode of action of sulfur fungicide depends on the release of hydrogen sulfide, which preferentially damages fungal and oomycete cell membrane and inhibits mitochondrial functions. Okay, they inhibit mitochondrial functions. This is not a very specific mode of action, and damage to plant is often observed. Damage to plant means phytotoxicity is often observed because of this sulfur fungicide. Okay, so sulfur is an example of a contact fungicide. Remember the example, that is best sprayed on a plant prior to a pathogen attack. So sulfur fungicide, it is sprayed. It can be sprayed afterwards, but it is best to spray prior to the pathogen attack as a preventive measure. The H2S means hydrogen sulfide attacks the pathogen spore on the surface and damages them to prevent penetration. It is widely used to control superficial disease like powdery and downy mildews on woody crops like apples and grapes, and also used on potatoes and cereals, especially in organic agriculture. Many metals and are toxic, and this applies also to copper, which combines efficacy with cheapness. It is absorbed by fungal and oomycete hypha and forms complexes with sulfitryl, hydroxyl, amino and carboxyl group in enzyme, causing inhibition and non-specific damage. Like sulfur, it is a contact fungicide giving a potential effect and is used against a wide variety of diseases. In addition to its use in Bordeaux mixture, it is sold as a series of formulations that balance the toxicity and persistence of the product. Copper is a general biocide and is toxic to plants and many other organisms. It accumulates in the soil to such an extent that many crops species fail to thrive. Copper is a table given. Okay, copper is a major fungicide for organic agriculture, and so extensive research is being undertaken to find a safer alternative. Tin is another metal still in use as a fungicide. So let's see what this table they are saying. So these are some of the basic substances used as fungicides, such as copper salt, sulfur lime, and calcium hydroxide. Copper salt, the target downy mildews, bacterial diseases. Okay, these are the examples of the crops. Lime sulfur, they use DM, anthracnose, black rot, exobasidium, phomopsis, leaf curl, and these are the crops. Okay, these are the examples of some basic substances used as fungicide. The next is current synthetic fungicides. Data from FRAC. Another question came to my mind. What is the full form of FRAC? Okay, go and search, because FRAC is an important thing that everyone should remember, because on the basis of their analysis and data, all the fungicides have been grouped. Okay. So code and target site such as RNA polymerase. Which fungicide they target RNA polymerase? They are the phenylamides. So phenylamides is the group name, and their their target site means it is similar to the mode of action or the site of action. Okay, they target the RNA polymerase activity and inhibit the function of the pathogen. Okay, so what are the examples of such a chemical of phenylamide? Is metalaxyl, benalaxyl. Now metalaxyl is a chemical used against which pathogen? Again, one question. I'll say the answer of this. It's Oomycetes fungi, they target metalaxyl. But nowadays what happens, or what is happening, metalaxyl is not that effective against Oomycetes. So new chemical has developed named Ridomil. Okay. So Ridomil is a combination of contact and systemic fungicide. And by the way, this phenylamide is a systemic fungicide. Okay. The next group is hydroxypyrimidines. So they target adenosine deaminase, and the example of such is bupirimide. Okay. The next, heteroaromatics. They target DNA or RNA synthesis. Fine. The next is methyl benzimidazole carbamates. They target beta tubulin assembly in mitosis. The example is carbendazim, thiabendazole, thiophanate, methyl. So see, these are some which are well known. They have highlighted it. See, they have highlighted it. The phenylamides, this is important. The benzimidazole, obviously, see, these chemicals are very well known. The metalaxyl, carbendazim, right? So then coming to this one, SDHI. I told you this SDHI for which group? Oxanthin group. Okay, they have this SDHI should have been here. So I would prefer the group name to be oxanthin group, in which the example will come as carboxyl, oxycarboxyl. Okay, that example is not given, oxycarboxyl, but yeah, both will come under the oxanthin group, and their target site is succinate dehydrogenase inhibitors. Okay, which is in mitochondria. The next important is this quinone outside inhibitor, quinone. These are the example of strobilurin group. Okay, so this is also one of the important one. Then coming to this quinone outside inhibitor. Okay, and then DMI, there was one short term, remember we were searching for it, DMI. So that is demethylation inhibitors that I already mentioned. And then yeah, there are various groups. Just see it through with their example and target site. Okay, in the table all this they have mentioned, but in a complex way. I believe this would have been a bit easier. Fine. Coming to dithiocarbamate fungicides. The dithiocarbamate fungicides were introduced in the 1930s and 1940s and are generally regarded as the first synthetic fungicides. Is it? Many remain in use today. Like copper and sulfur, they are immobile products that remain on the leaf surface, but they are also used to treat seeds. The most successful product is mancozeb, which is a complex mixture of dithiocarbamate backbone with manganese and zinc ions. Like the sulfur fungicides, their MOA means mode of action is to react with HS groups. HS group means sulfur dioxide or hydrogen sulfide, sorry, hydrogen sulfide groups in pathogen proteins, but they are much less phytotoxic other than sulfur products. They are broad spectrum products with major markets in controlling Venturinia scab, grape tobacco and hop downy mildew and potato late and early blight, but are inactive against the powdery mildews. The next major development was the introduction of phthalimid fungicide Captain, yeah, and Folpate by Standard Oil in 1952. Folpate remains in widespread use. Like the dithiocarbamate, they act as sulfitryl reagents inactivating SH groups in enzymes in cell which they contact. The phthalimates are preventive fungicides for a wide range of fungal and mycetes pathogen. They control downy mildews, botrytis, potato leaf blight and apple scab, and are used extensively as sprays, root dips and seed treatments. Such the banning of chlorothalonil in Europe in 2021, Folfate has assumed as a greater role as a mixing partner for many systemic fungicides, increasing the spectrum and protecting against fungicide resistance. After dithiocarbamate, the next is systemic fungicide methyl benzimidazole carbamate and the emergence of resistance. The introduction of methyl benzimidazole carbamate fungicide in 1969 marked the start of a new phase of fungicide industry. Benomil and thiabendazole were followed by trifenate in 1971 and carbendazim in 1974 and were first fungicides with systemic activity. They were absorbed from the foliar tissue and roots and transported via xylem flow. The ability to move systematically through a plant gave these compounds the ability to control pathogens that had already penetrated the tissue of the plant. That is, they are curative as well as preventive. So these are curative as well as preventive. These give a substantial advantage over the older contact and immobile contact fungicides and contributed to a sustained period of yield growth. Yes, because of this curative and preventive measure, they have given the upper hand of being more beneficial. Okay, all right. Many examples have been mentioned. Moving forward, the next is, look, I'm scrolling everything, but if I read word by word, that it is not going to finish. Any chapter of gene Agrios is very vast. Sterol biosynthesis inhibitor. So C14 demethylase multiple mechanisms of resistance. The second major class of systemic fungicide to be introduced was the C14 sterol demethylase inhibitors, often known as demethylation inhibitors, or informally as the triazoles. Okay, so I have given you the examples, right? The triazoles, this triazole group's mode of action is sterol biosynthesis inhibitor. Okay, so they inhibit the sterol biosynthesis. Now, sterols are major components of cell membrane in all eukaryotic organism and function to maintain membrane fluidity and integrity. The main sterol in animal is cholesterol. The plants have more varied group called the phytosterols. The pathogenic mycetes do not synthesize their own sterol but obtain them from their plant host. As a result, fungicide which inhibit the sterol pathway are inactive against mycetes diseases. Okay. The main sterol in fungi, okay, remember this, the fungicide which inhibit the sterol pathway are inactive against mycetes disease. So there are many mycetes diseases such as what? Pythium, Phytophthora, they're the pathogen I meant to say. So yeah, the diseases caused by them. So this sterol fungicide will be ineffective against such diseases. The main sterol in fungi is ergosterol. But some species such as Venturi, powdery mildew and rust have variants such as 2,4-methylene cholesterol. The biosynthetic pathway from acyl CoA is broadly similar in all fungi, but details of the intermediates vary as well as the order in which they are synthesized. Acetyl CoA is converted to quilin and then in multiple steps. 5 steps in the pathway have been exploited in the design of fungicide to control both agricultural and clinical diseases. Inhibition of the pathway not only depletes the amount of the final product, but it also forces the accumulation of intermediates which are themselves toxic. The dual action gives sterol biosynthesis inhibition their potency. The 5 states are defined by the genes in the model is Saccharomyces cerevisiae, example ERG1, 7, 2, 24, 27, but the most important is ERG11, which encodes an enzyme called sterol C1 for alpha demethylase. It is a heme containing cytochrome P450 complex that function to remove a methyl group from the carbon 14 of the steroid. In filamentous fungi, the enzyme and gene are called CYP51. All fungi have a paralog called this CYP51B. Other species have an additional gene called CYP51A, and often these genes are duplicated. Inhibitors of this encode sterol demethylase are the most important class of agricultural fungicide in terms of cells number of derivatives actives of different actives and impact on disease control. Oh my god, there are many things in between that I believe, we should not get that deep into the insights, the anatomy of a chemical preparation, how they are done. So for now this is not such important if we want to understand the topic. Yes, if we want to go deep and do some research on them, or we want to develop some new fungicide, new chemicals, you are most welcome to study everything. Okay, many more things have been mentioned here. Moving forward. Okay, this topic is vast. The next, let's see about quinone outside inhibitor resistant risk factor. The mitochondrion is the major source of energy in the cell, contributes to intermediary metabolism and controls apoptosis. It has proved to be a vulnerable target for fungal and oomycete pathogens with as many as 8 different classes of fungicides. The most important are SDHI and QOI, means the quinone. Okay, inhibitors I meant. So QOI fungicides were introduced from 1996. They bind to the quinone site in complex 3 of the mitochondrial electron transport chain and have potent activity combined with remarkable safety and spectrum. QOI fungicides control all types of fungal and oomycete pathogen but have exceptionally low levels of phytotoxicity. They are systemic and are mainly used as foliar spray and as seed treatments. They possess curative activity but are best suited for preventive roles. So quinone outside inhibitor, they are best suited for preventive role rather than curative. Okay, remember this. Next is succinate dehydrogenase inhibitor. Another mitochondrial complex that has proved to be an important fungicide target is complex 2, which is also known as succinate dehydrogenase. The SDH succinate dehydrogenase complex is made up of 4 subunits, SDHA to SDHD, means A, B, C, D. each encoded by a different nuclear gene. Electrons from succinate are transferred via FAD bound to SDH-A via 3 iron sulfur cluster in SDH-B to ubiquinone at the Q site formed by SDH-B, C and D. The fungicide bind the Q and the block transfer of electrons. The first SDH inhibitor were the carboxyls. Yeah, this is what I was talking about. Introduced as long as ago by as 1966. So this was the question that I was asking. Okay, so this introduction was made by whom? The first SDH inhibitor carboxyl was introduced by whom? Okay, they were only used to control basidiomycete seed-borne disease such as cereal smut and buns. A second generation of actives was released from 2003, which with a much broader spectrum. Oh, what happened? Okay, sorry. Yeah, a second generation of actives was released from 2003, which are much broader spectrum, including ascomycetes, fungi, but not oomycetes. Compounds such as boscalid and bixafen revealed the potential of these compounds, and since then another 10 actives have been released. SDHI have a simple carboxymide group linking two aromatic rings. They are used to control seed-borne and foliar diseases in a wide range of crops. The group has 11% of the market and is the third largest group after DMIs and quinones. The newer SDHI are generally sold in mixtures with other MOS. Fungicide for oomycetes diseases. Oomycetes pathogens present a contrasting challenge for the fungicide industry, as they are not controlled by methylbenzimidazole, DMI, dimethyl inhibitor, or SDHI, succinate dehydrogenase inhibitor activates, but they are sensitive to quinones inhibitor. Okay. In addition to the quinone outside inhibitors, two major classes of fungicide are used: the phenylamides and the carboxylic acid amides, as well as 13 minor groups. These compounds are still almost universally called fungicide, despite the recognition that oomycetes are very distantly related to the true fungi. Occasionally they are called either oomycetes or oomyceticides. These compounds are partially important in the control of potato late blight and downy mildew of grapes. The most important systemic fungicides specific for oomycetes are the phenylamides, metalaxyl and benexyl. They have been in use since 1979 and are used as protectants and curatives in seed treatments and in root and foliar application. Sells an approximately $500 million. Only one of the enantiomers of benaxyl and metalaxyl is active, and these have been separated and are now marketed as Kyraxyl and mefenoxam respectively. Okay. Now we will know about biological control agents. A small but fast growing sector within CPP is in sales of biological control agents. Okay, BCA are living organisms that control pest and pathogen of crops. The largest sector of BCA market is for products designed to control insects, but a significant fraction is deployed to control diseases. One significant factor is the fast growing of this market is to support crop protection in agriculture, that is marketed as organic or biological. The concept behind biological control replicates the competition between microbes on and in a plant, that is a feature of natural ecosystem. If the BCA survives and reproduces on the plant, it can provide prolonged disease protection that may even extend it from one growing season to next. The discovery of useful BCAs normally began with a screen of microorganisms, mycetes, fungi, bacteria, viruses that kill or at least inhibit the growth of the pathogen. If possible, potential screens are tested in vitro for activity against the test pathogen, but often it is necessary to apply the BCA directly to the infected field. Useful BCAs must undergo battery of tests to support regulation both with national pesticide regulators and with organic certifiers. A major problem of BCA supplier is to establish conditions for the bulking up, formulation, packaging and delivery up to the farmers. This tends to be much more complex than for synthetic pesticides, as a living organism is involved. Definitely, as a living organism is involved, which means packaging it and how long it will live. The shelf life depends much more important rather than the chemical ones. Okay. Now, defense induction. All plant species are resistant to most pathogens. Remember this line. All plant species are resistant to most pathogens. Recognition by the plant that is being attacked normally leads to the induction of a generalized defense response, characterized by the synthesis of anti-pathogen molecules, cell wall strengthening and hypersensitive cell death, and so-called PAMP, MAMP, PTI. Okay, we know these terms, read in chapter 5 while going through plant immunity. Okay, so yes, all plant species are resistant to most pathogens. That means what we can say? Disease is an exception. Plant is naturally resistant to most of the pathogen and disease is the exception. Okay, because for a disease to occur, there are multiple factors that needs to coincide all together, and that's when the disease triangle and then tetrahedron and everything comes up, in which all the parameters should coincide, and in that position the disease will occur. Fine. Induction of PTI by CPP can therefore enhance disease control. Several synthetic fungicides in FRAC class P also work in this way. The BCA Tegro, which is a strain of Bacillus subtilis, control a range of disease in vegetables and potato by inducing a defense response. Hyperparasitism. Hyperparasite, BCA, are specific disease of the pathogens that cause disease in plants. When the host is a fungus, this is called mycoparasitism. Powdery mildew fungi are host to the fungus Ampelomyces quisqualis. So this is one example that everyone should remember. What is the hyperparasite used as a biocontrol agent for or against powdery mildew? That is Ampelomyces quisqualis. Okay, which is formulated as AQ10 and used to control disease in protected crops and vegetables. The fungus Coniotherium minitans is a specific pathogen of the Sclerotia, Sclerotinia, Sclerotium, the cause of disease in many crops such as canola. It is marketed as Contas WG. Broad host free-living fungi from the genus Trichoderma group are used in at least a dozen preparations to control damping of disease in a broad range of situations. It is likely that Trichoderma species combine several MOAs, including antibiosis and competition. Several plant viruses are controlled by bacteriophage such as agriphage. I hope you know about bacteriophages. Okay. Next, competition. If the BCA grows on or in the plant and competes with the pathogen for water, nutrient or simply species, the MOA is called either competition or exclusion. MOA is what till now? This is mode of action. So mode of action, what will happen in case of biocontrol agents if they target the or if they compete the host pathogen for water, nutrition, supply. So there will be this race who will win. So in this case, the MOA will be competition or exclusion. This is the most common MOA of BCA. A common theme is to screen for or select strains for a pathogen that lack a key virulence function. For example, Agrobacterium radiobacter competes with Agrobacterium tumefaciens but does not induce galls, or strains of Aspergillus and Fusarium that do not produce mycotoxins that compete with toxigenic pathogens. Next comes antibiosis. Other BCAs control pathogens by screening molecules that directly interfere with the growth of the pathogen. Gliocladium virens produce the molecule gliotoxin that is thought to be the cause of the control, but the details have not been determined. The product Pristop or Primostop has Gliocladium virens and controls soil-borne disease of ornamental vegetables, turf and trees. Coming to botanicals. Botanicals are the term used for pesticides that are natural products that are sold as products to be applied to crops and crops to control diseases. They are normally derived from the plants, and as long as they have not been chemically converted, they are permitted in organic agriculture. They undergo stringent safety and efficacy testing prior to registration, just like synthetic pesticide. Several products work by exerting directly toxicity and hence are comparable to the early contact fungicide being used as preservatives only. Regalia is a substitute anthraquinone island from Rhinotria saccharinensis and controls a wide range of bacterial and fungal diseases via induction of defense. Next come antibiotics. Antibiotics means that work against bacterial diseases, right? So antibiotics are defined in the plant pathology as compounds that control bacterial diseases. Only a few compounds are in current use, mainly because they are very prone to the evolution of resistance. The main example is streptomycin, produced by Actinomyces streptomyces gracius. Now you know streptomycin is one such example of antibiotics that streptomyces is one such source through which more than half of the antibiotics are made from the source called streptomyces, and streptomycin is produced from specifically streptomyces gracius. Okay, it binds to the bacterial ribosome and prevents protein synthesis. Okay, now here comes again the mode of action. So there, even in antibiotics, there are several mode of action. Fine. So there are various groups called beta-lactams, and then tetracyclines, and then neomycin. Many are there. Okay. So among those, streptomycin comes under, if I'm not wrong, it will come under polyglycosides. Okay, polyglycosides. No, sorry. Streptomyces, the chemical group for streptomycin is aminoglycosides, not polyglycosides. Very sorry. So their mode of action, like I say, it will be protein synthesis by binding with 30S ribosomal subunit. Now similar thing you can see in the group called macrolides. Example of macrolide group can be erythromycin or carbamycin, and they will again inhibit, means prevent the protein synthesis, but in that case they will do that by binding with 50S ribosomal subunit. Okay, so there are many mode of actions for antibiotics and their chemical groups such as beta-lactams, aminoglycosides, macrolides, rifamycin, so on and so forth. Beta-lactam is one of the important ones, because under beta-lactams penicillins come, and everybody knows about penicillin. Penicillin is the first antibiotic, right? Okay, by Alexander Fleming in 1928. Again, if I'm not wrong, I hope it is 1928. Now that penicillin, in that case, the mode of action is they inhibit the murin synthesis. Okay, so for penicillin, mode of action is murin, they inhibit the murin synthesis. For streptomycin, they inhibit protein synthesis by binding with 30S ribosome. And for erythromycin, same, they inhibit protein synthesis by binding with 50S ribosome. So yes, again, they have different mode of actions. Now, streptomycin is sold as agrimycin and phytomycin, and as a spray shows activity against a broad range of bacterial plant pathogens causing spots, blights and rots. Streptomycin has also been used as soil drench. Example in the control of geranium food rot caused by Xanthomonas species, as a dip for potato tuber species used for seed against various bacterial roots of tubers, and these seed disinfected against bacterial pathogens of beans, cotton and so on and so forth. Next, coming to nematicides and insecticides. Okay, many of the nematicides are broad spectrum volatile soil fumigants that are active against not only nematodes but also insect, fungi, bacteria, weeds, seeds and almost anything else living in the soil. Several newer chemicals are non-fumigant, granular or liquid substance actively mostly against nematodes and insects. Next comes not next, under this it will come organophosphate nematicides. Okay, and then isothiocyanates and carbamates. Okay, so organophosphate nematicides include the insecticides Forate, disulfoton, etoprop and etc. and few others. Many of the organophosphates were developed initially as insecticides. However, they were taken up and are distributed systematically through the plant and are effective nematicides. Okay, they are available as water soluble liquids or granules, have low volatility, can be applied before or after planting, or are effective only against nematicides. Most have minimal or no activity against soil fungi. Okay, these nematicides inhibit the neurotransmitter enzyme cholinesterase and result in paralysis and ultimately death of the affected nematodes. Coming to isothiocyanates. So this includes metam and dazomate. They are active against nematodes, soil insects, weeds and most soil fungi. Metam, sodium and Vorlex are applied by injection, incorporation or irrigation into the soil at least 2 weeks before planting. Basamine is a granular product. They all act by releasing methyl isocyanate, which inactivates the SH group in enzyme. The last that will come under nematicide, that will be carbamates. So carbamates include aldicarb, carbofuran, oxamil and carbosulfan. They are active against nematodes and soil insects as well as some foliage insects, available as granules or liquids of low volatility. They are easily soluble in water and can take up and translocated systematically by the plant. They are incorporated into the soil by disking before or after planting. They act by inhibiting the enzyme cholinesterase, causing paralysis and death of the affected nematodes and insects. Method of application of chemical for plant disease control. Chemicals used to control plant disease are applied directly to plant or to the soil with the help of various types of equipment. Chemical applied as sprays or dust on the foliage of plants are usually aimed at the control of fungal and oomycetes disease and to a lesser extent control of bacterial disease. Some fungicides and bactericides are protectants and must be present on the surface of the plant in advance of the pathogen to prevent infection. Yes. Their presence usually does not allow fungal spores to determine, or the chemical may kill spores upon germination. Contact of bacteria with bactericide may inhibit their multiplication or cause their death. Systemic fungicides can be absorbed by parts of the leaf tissues and translocated internally into the whole leaf area. Fungicides applied as sprays are generally more efficient in creating a protective residue layer on the plant surface than when applied as dust. Neither dust or spray stick well when applied during rain. And definitely, if rain will come, how they will stick? how they will be effective. Okay, sometimes other compounds, example lime, may be added to the active chemical to reduce its phytotoxicity and make it safer to the plants. Use of lime. Compounds with a low surface tension called surfactants are often used to fungicide added to fungicides so that they spread better. Now you know these are some of the methods. That's how the chemicals are applied for plant disease control. They can be used as dust or spray or as a surfactant or stickers. So these are the way or the methods by which the chemicals are applied. See examples of dusters, sprayers, fumigators. Okay, let's see what is there next. Is seed treatment. So seeds, tuber, bulb and roots are often treated with crop protection products to prevent their decay after planting or the damping off of young seedlings. These chemicals may control pathogens carried on seeds, tuber and so on, or existing in the soil where they will be planted. Since the mid 1970s, seeds have been treated with systemic fungicides or in order to inactivate pathogens in infected seeds, or in order to provide the foliage for the developing plants with systemic protection against the pathogen. Okay, chemicals can be applied on the seed as dust or thick water suspensions. This is how chemicals can be applied to seeds. Okay, mixed with the seeds. This seed can be soaked in water. The seeds can be soaked in water or solvent solution of the chemical and then allow it to dry. Tubers, bulbs, cones and roots can be treated in similar ways. But treatments are effective mostly when the chemical is applied to protect such organs when they are healthy from infection in the field. Means this can be used, the seed treatment is used as a prevention rather than eliminating pathogens that have already infected the organs. Understood? Yes, it is used as a prevention rather than the ones in which the seeds are already infected by pathogens. Okay. In treating seeds or any other propagative organ with chemicals, precautions must be taken so that their viability is not lowered or destroyed. At the same time, enough chemicals must stick to the seed to protect it from attacks of pathogen. When the seed is planted, the chemical diffuses into the soil and disinfects a sphere of soil around the seed. In this way, the new plant will grow without being attacked as their particularly vulnerable period of growth. Understood? Tactics in the use of CPPs. There are three main strategies used by farmers to decide when to apply CPPs. The first is to apply when the crop reaches certain growth stage. The second is to wait for a certain threshold level of disease, and the third is to use as a disease prediction model. We will start with the first. The first is disease threshold strategies. I know this session is going a little longer, but I'm willing to complete this chapter in this single one. Yesterday we did half, not half, first 30%. Today we are covering the total 70%. So still few pages are left. Stay with me. If the disease threat is intermittent, it is advantageous to limit CPP's use to when the disease level are high enough to justify the cost. This sounds all well, but in practice, it may be very difficult for a farmer to purchase, receive, dispense and spray a fungicide in response to an unexpected change in the weather. A much more likely scenario is that a farmer schedules a series of sprays for the growing season but makes the decision to omit one or more sprays if the disease development or date and weather focus do not justify the spray. The fungicide will be used on farm on a future or traded with a neighbor. This strategy requires the farmers to monitor the crop for disease. Visual symptoms are clear, but it is also possible to use either molecular detection or imaging for a more sensitive or more timely diagnosis. Okay, more examples are given. Moving forward. Then disease threat models. Okay. Weather forecasting is improving all the time and can now give reliable predictions for some days in the future. Most diseases require specific rainfall, temperature and humidity in order to flourish. This data can be linked to the forecast and used to give early warning to disease, likely disease. In many countries, extension and CPPs resellers generate mobile or cell phone alerts to farmers to advise. Okay. Disease threat model. So yes, you have heard of disease forecasting model. Definitely, you have heard of disease forecasting model. We have also covered it in our prior chapters. So, for example, few of the disease require specific rainfall, temperature, humidity, right? So if you study about Dutch and Mills rule, Dutch for late blight of potato and Mills rule for apple scab, there you will see there are specific temperature and the dew temperature that is given, soil temperature, okay, then the overall temperature required, and if that is found, then the disease is most likely to occur. And nowadays what is happening, the farmers or the researchers, they are setting mobile alerts or cell phone alerts to advise them when to spray crops and what to do. There are various sensors, when to do irrigation, when to spray for chemicals, and when to harvest. All these things nowadays, these mobile apps, they are regulating. Okay. Resistance management. Like genetic disease resistance, pathogens have evolved to overcome the protective effects of CPPs. Although the early multi-site fungicides have proved more or less immune to resistance, single-site fungicides and antibiotics have proved to be vulnerable to the evolution of resistance. What does this mean? I mentioned contact fungicide and systemic fungicides. Now, contact fungicides are multi-site fungicides, whereas systemic fungicides are single-site fungicides. Now the multi-site fungicides have proved more or less immune to resistance because there are multiple sites of action, so it's difficult for pathogen to be resistant to that. Okay, but single-site fungicides, what happens? Single-site fungicides, they have only single site of action, so they are more vulnerable to evolution of resistance. That is what they are saying. Okay. The evolution of resistance to antibiotics and to the PA, BMC, DMI, SDHI and QOI, these are the fungicide groups, has been observed over the last 50 years. During this time, observational studies, experiments and modeling have been applied in order to determine the best strategies for resistance management. The goal is to reduce the risk that resistance will evolve and to prolong for as long as possible the effective life of a fungicide. Fungicide work because they limit the growth of rate of pathogen. There is therefore a selection pressure for the pathogen to evolve resistance. The goal of fungicide resistance management is to what? Minimize the magnitude of the selection pressure. A consensus has emerged around a series of ideas known as governing principles, first proposed by Milgrom and Fry. These principles can be summarized as 1. Principles 1. The use of minimized dose of fungicide that gives adequate disease control. In most cases, this dose will correspond closely to the dose that gives the best economic return. The second, ensure that the pathogen is exposed to the fungicide for the minimum time consistent with adequate disease control. The concept has led fungicide companies to place limits on the number of applications that a particular fungicide can be applied in each season. Third, the fungicide with different mode of actions, either as mixtures or in successive applications. The idea is that a pathogen that has evolved resistance to one fungicide is still likely to be sensitive to a second. The second fungicide controls the disease and prevents the spread of the resistant population. The strategy relies on both fungicide being effective. If either fungicide suffers from resistance, it must be replaced in the mixture and alteration. Also, the strategy is under threat from the evolution of MGR, and most, if not all, fungicide classes are compromised. Number 4, the monitoring of resistance is highlighted. In the past cases, the fungicide resistance were discovered only after a damaging outbreak of disease. By then, the resistance pathogen population has grown to dominate, and future use of the fungicide is severely compromised. It is much better to detect fungicide resistance early. To plant withdrawal of the fungicide will allow the sensitive population to recognize the area. And the last one is integrated pest management, the most important and effective strategy used to employ all suitable methods, especially genetics and agronomy, to keep distance to keep disease levels in check. These are all the principles. Okay, all the principles that can be used as disease management, resistance management. Okay, the next is integrated control in a perennial crop. Then we have integrated control in annual crop, and let's see what more is there. Comparison between the 4 methods of protection. This has come to an end. Okay, so just a few more minutes with me. Integrated control in a perennial crop. In an integrated control program of an orchard crop such as apple, peach or citrus, one must first consider the nursery stock to be used and location where it will be planted. If the fruit is susceptible to certain viruses, molecules, crown gall bacteria or nematodes, the nursery stock must be free of these pathogens. Okay, this is the first thing that needs to be checked. Then stock free of certain viruses and other diseases can be usually be bought from selected nurseries. So first we will check from the nursery whether the stock is free from certain pathogen. One such is virus. Yes, so first we will check that, and then it will be brought from selected nurseries whose crops are inspected and certified. If the possibility of nematodes on the roots on the root stock, the stock must be heat treated. So this is one of the method that can be used if nematode is found in that root stock. The location where the trees will be planted must not be infested with oomycetes or fungi such as Phytophthora, Armillaria or numerous nematodes. Okay. If it is, the field should be treated with fungicides before planting and varieties grafted on rootstock. First we will check the nursery for the healthy rootstock, and then we will check the location where this rootstock will be planted. So everything will be well checked before planting. Okay, the drainage of the location should be checked and improved if necessary. Finally, young trees should not be planted on sites previously occupied by similar crops, particularly by the latter where diseased or or next to old trees and are infected heavily with canker, fungi and bacteria, insect transmitted viruses and molecules, pollen transmitted viruses or with other pathogens. Once the trees are in place and until they begin to bear fruit, they should be fertilized, irrigated, pruned and sprayed for the most common insect and diseases, so they will grow vigorously and free from infections. Later on, when the trees bear fruit, the care should increase, as should the vigilance to detect and control disease that affect any part of the tree. Any trees that develop symptoms of a disease caused by a systemic pathogen should be removed as soon as possible. Disease control measures in an orchard may begin in the winter when weak, diseased or dead twigs, branches or fruits are removed during pruning operations and are buried and burned. Okay, so fine, the fruit has come, and then further management should be taken. Done. Now integrated control in an annual crop. In an integrated control program of an annual crop, one must again start with a healthy seed that carries as much as useful genetic resistance as possible. Next, planting materials such as potato seed tubers or the true seeds of cereals, legumes and oilseeds should be from source where they are tested for pathogens. Nutshell, healthy seeds needs to be taken. Fine. And then where the field, then in the annual crop, in the perennial crop, the location is tested for rootstock and the location, and then here similarly the healthy, they should choose unhealthy seeds, and then the field should be free of pathogens. It is best to rotate one crop with another, as unrelated crops will usually reduce the population of specific pathogen. The soil may have to be treated with a fumigant if it is known to be infested with the rootstock or other nematodes. If the disease occurs regularly year after year, the grower should start spraying with the appropriate fungicide as soon as the disease appears. Insecticide sprays control insect may reduce the spread of some viruses. Using weather data to focus disease appearance and development can help in spraying at the right time and in not wasting any sprays. Sprays must continue throughout the growing season as needed and as the weather dictates. Each one of these measures must be used for the best results, and the routine use of each of them makes all of them much more effective. Now the last one, comparison between the 4 methods of crop protection. Now what are those 4 methods of crop protection? Finally, it is biosecurity, agronomic, genetics and CPP crop protection products. So rather than reading this paragraph, we will directly come to this table. Okay, so cost of farmer. For biosecurity, zero cost to farmers. For agronomy, limits the growing of the most profitable species to once every few years. Cost, time and diesel. Yes, because it's zero cost to farmers. Why? Because in biosecurity, what happens? They prevents the entry of pathogen into the farm. So there is no headache of farmer. So zero cost to farmers. But in agronomy, what happens? The pathogen is already in the field, and what the work of farmer is to, they can do crop rotation or something, in because of which what happens? The most important crop, the most the most profitable crop is not grown year after year, so it cost them time and profit. Okay, it cost them profit. Fine. For genetics, what happens? Resistant seed may be more expensive, yes, or may come with yield penalties in the absence of disease. Okay, so for CPP, from $5 to $1,000 per hectare. So this is the cost if the farmer do such practices. They can't do this practice, but what are the cost of the farmers in case of agronomy, genetics and CPP methods? Okay, now spectrum only applies to disease not yet in the vicinity of the farm. Fine. For agronomy, potentially provides complete control of root disease and modest control of rain splashes disease, little impact on airborne diseases. Okay, I hope these sentences are very simple. With this you can understand. Limit to the resistance gene carried by the cultivar. It is limited to the resistance gene only. Okay. For CPP, most fungicides control is wide range of pathogen, and a few are very broad spectrum. Products specific for powdery mildew and botrytis are available. So in general, for chemicals, there are various chemicals which are broad spectrums, means it is effective against lots of pathogens. Okay, and few chemicals are there which are specific. Okay. Next is longevity, entire season, because they are preventing the pathogen to enter into the field. So yeah, entire season is saved. The agronomy, impact declines through the growing season. Okay. For genetics, although some gene operates only as seedling or in adult plants, protection throughout the season is generally achieved. Very nice. Extreme temperatures can reduce the effectiveness. Okay. For CPP, provides control only for a maximum of a few weeks or each spray. Rainfall events reduce the longevity of protection. We have studied this in the paragraph. Okay. Next, longevity between season. Incursions eliminate the protection. Must be reapplied each season. Disease resistance gene can be durable, but most fail or at least decline in efficiency in few seasons. And for CPP, resistance has developed for many classes of fungicides, reducing the efficacy, requiring either the use of higher doses or a different fungicide. Then ecological sustainability. For biosecurity, it protects the wider environment beyond the farm. For agronomy, plowing and green breach control can damage ecosystem. For genetics, no direct impact on ecological sustainability. Why? Because they work on genetic basis. Okay, they work with genes. And the last with CPP, highly regulated for off-target effects but negative impact of insecticides, and to a lesser extent fungicides, and are inevitable to a smaller extent. Okay, that was the last topic. Fine. That's how this big chapter, why big chapter, because in this one chapter they have summarized, they tried to summarize everything, all the management techniques. I hope it was clear, yesterday's session and today's session all together. Okay. So in general, if you have heard in GN Agrios, the first 10 chapters are important. So yeah, the first 10 chapters are finally finished. So you can revise these 10 chapters again and again. But yes, there are more chapters, but it will come eventually. In the further chapters, you will see about diseases. Okay. Diseases of fungi, the bacterial diseases, viral diseases. So all this will come up in the following chapters. But for all of us to understand about plant pathology, first to chapter 10 is the most important chapters that everyone should study before heading to any competitive exams. Fine. Or if anybody wants to understand plant pathology. So till then, till this chapter 10, revise again and again and again and again. And the further chapters will come up after a week or two. Okay. So yes, it will definitely come. I promise the further chapters will definitely come. But 1 to 10 is the most important one. Okay. Good luck. I'll not extend this video much longer because I hope it's a lengthy one. You study well, keep learning, keep growing, and all the best whoever is appearing for exams. Till then, keep learning, keep growing. I have repeated it twice. Okay, now bye.